Hello students, today we will start with new topic that is about translation. Translation can be defined as a process in which there is polymerization of amino acids for protein synthesis or simply protein synthesis by using mRNA and the genetic information that is present on mRNA. So there are some requirements for the process of translation. What are those? Those requirements are mRNA, tRNA, ribosomal RNA, which is one of the constituent of rRNA, means rRNA in form of ribosomes. Next is ATP molecule, because it is necessary for formation of peptide bond between tRNA and amino acid. Fifth one is enzyme called amino acyl synthetase. Amino acyl synthetase. There is another element that is required, that is magnesium ions. So these all are considered as the requirements of translation process. If we go for step-by-step representation, first point we have to observe that peptide bond formation requires energy. We will see some point or explanation regarding this statement. So the basic structure of an amino acid will be one carboxyl group, one amino group, another amino acid, one carboxyl group, one amino group, and one hydrogen. So these are considered as the basic structures of amino acid. Now here the hydroxyl group of this carboxylic acid and hydrogen of the amino group, both of them will be removed in the form of water. When hydrogen or H plus ion from ammonia is getting released, it means that the electrons are also released here. So some energy is also released. By utilizing that energy, the peptide bond is formed between two amino acids. Now the structure will be looking like, so one water molecule has gone out by the process of dehydration. So there will be a bond between carbon, nitrogen, and also here this total bond between nitrogen, carbon is called as peptide bond. Peptide bond, that is between two amino acids. Here it is a process of polymerization of amino acids to form into a protein. We can also include here to form into a polypeptide chain. It means that if many number of such peptide bonds are present in one long chain, then we can use a term like polypeptide chain. Now we'll see the steps that are included in translation process. So first one is activation of amino acids that are present in cytoplasm. Basically, ribosomes contain many number of amino acids, but they are inactive in form. Those amino acids during this protein synthesis has to get activated. So activation of amino acids. This step includes ATP. So here ATP is required. Next, enzyme called amino acyl synthetase. It is also required for the formation of a complex, that is amino acid ATP enzyme complex. So we get now amino acid ATP enzyme complex. Now this total complex causes activation of the amino acid. Activated form of amino acid ATP enzyme complex. Initially, they are all bounded with each other. Later on, they will be activated in form. Now, the amino acid that is initially involving in the translation or translation process or getting activated, it can be methionine. So in eukaryotes, the first amino acid activated or involved in process of translation is methionine. Methionine. We know that there are 20 number of amino acids that are responsible for protein synthesis. Next, in case of prokaryotes, the first amino acid that is involved in translation is formilated methionine. We call it as F-met, or this is just symbol to represent. This is called as formilated methionine. Formilated methionine. Now, this is about first step, that is activation of amino acids. Here, this ATP will be broken down and energy is released. Then, when that ATP is broken down, then the compound that is attached to the amino acid enzyme together complex will be AMP, means adenosine monophosphate. Next step is charging of tRNA. Second one is charging of tRNA. This can also be called as amino acylation of tRNA. Amino acylation of tRNA, or we can also use with another sentence, that is transfer of activated amino acid onto tRNA. Transfer of activated amino acid onto tRNA. Now, this amino acid is already activated with the help of enzymes like amino acyl synthetase and also the energy giving compound, that is ATP. The activated form of amino acid from, so amino acid I am writing with small a, with AMP form of ATP when it is broken down, and enzyme, now when they are binded with tRNA, gives now a complex, that is amino acid AMP enzyme tRNA complex. Now, this all will be now causing the releasing of this enzyme. Here, while framing such compound, there is a catalyst that is supporting in formation of peptide bond between tRNA and also amino acid. That this point is according to NCERT. So, the peptide bond between tRNA and amino acid is catalyzed by a catalyst. What might be the catalyst? Here we can consider catalyst as a ribosome. Means ribosome is acting in the form of a catalyst here. There is a separation of this AMP and also enzyme from the total complex to form an amino acid tRNA complex. The AMP and enzyme, that is amino acyl synthetase, are separated from amino acid, AMP, enzyme, tRNA complex forming into amino acid tRNA complex. Between these two, the formed bond is peptide bond. Next step is protein synthesis or addition of amino acids one to each other. Third step is assembly of amino acids. So here on ribosomal units. This ribosome, it has two different subunits. One is larger subunit, one is smaller subunit. Smaller subunit has the site to bind onto mRNA. Has two subunits. Since we are talking about this total process in eukaryotes, let's think that this ribosome is of ATS type. Means it is having two subunits according to this point, that is 60S subunit, 40S subunit. So here the smaller subunit has the site to bind on to mRNA. What about larger subunit? Larger subunit has two special sites that accommodate the amino acid along with the tRNA and supports in formation of peptide bond between two amino acids. Larger subunit has two sites, that is A and P site. A site is termed as amino acylation or amino acid binding site. P site is nothing but peptide bond forming site. So now, if this is the mRNA end will be 5' to 3' direction. If you take, the smaller subunit will initially bind onto mRNA at one specific place where protein synthesis or translation has to be initiated. Later, the larger subunit will come and bind onto the place where smaller subunit has fitted. It is having two special sites, that is A site and P site. After formation of peptide bond, there will be exiting of the empty tRNA without the amino acid. So this is smaller subunit. This is larger subunit of ribosomes. Now what about tRNA? tRNA, we know that it is having some special structure like this. This end is called 3' end with OH. This is called 5' end. This is called DHU loop. This is called T-Psi-C loop. This is called anti-codon loop. This is called variable arm. This DHA loop, it helps in finding of amino acid, means identification of amino acid which has to be binded onto tRNA and gets activated. So this area or this arm helps in identification of activated amino acid. Then T-Psi-C loop. This helps in identification of the location of the ribosome for the process of translation. Helps in locating larger subunit of ribosome. Anti-codon loop. This one helps in reading the codons which are present on mRNA. Helps in reading codon sequence on mRNA. So, for suppose, if mRNA is having some sequence like AUG, if mRNA is having a codon sequence like AUG, then the anticodon will be uracil, adenine, cytosine. Then it will attach that amino acid which is nothing but methionine here. But the same, I will write another method. If the codon is uracil, adenine and cytosine on the mRNA and the anticodon is AUG. Basically, we read AUG as an initiating codon, but that is correct only when that is present on mRNA. Now we are writing adenine, uracil, guanine in the form of anticodon. Now the amino acid which is binded on to the 3' end of tRNA, it varies. It will be tyrosine. So whenever we are locating the anticodon and the codon on mRNA, it should be very specifical by saying is it the codon or anticodon. Because all the time we read AUG codes for the starting methionine, also called as initiating codon, etc. But let's first observe where it was written. Is it written on mRNA? Then it is correct. But if it is written as in case of anticodon loop of tRNA, then the amino acid that will be binded onto the 3' end is nothing but the tyrosine. And here 3' end will be having special codon sequence, that is CCAN. We have to read this CCAN as it is. We should not read like ACC because tRNA should be read from 5' to 3' direction. So here the codon sequence which are present on the 3' terminal end should be read as CCA sequence is present on 3' end of tRNA. If we see the diagrammatic representation of formation of a protein or polymerization of amino acids, mRNA, it will be read from 5' direction to 3' direction, and the smaller subunit will first recognize the initiating codon, that is AUG, and will come and fit there in case of eukaryotes. Now, AUG codon is very similar or nearby the P site, means peptide bonding site of the larger subunit of this ribosome. Now, tRNA already in the cytoplasm, it will be in the activation process, means it is getting activated by the amino acid ATP complex. So here will be anticodon for the tRNA. Here activated amino acid is ready to bound onto the 3' end of the tRNA. Now here we are seeing the total translation unit as mRNA, ribosomal both subunits smaller and larger, and also activated tRNA. The next step, this tRNA which is activated now along with anticodon UAC, that is coding for amino acid methionine, will come and bind onto the P site, not to the amino acylation place. Because the codon which is present, which is responsible for initiation of a protein, is present very similar to that of, very near to that of P site, that is peptide bond forming site. So this is 5' and this is 3'. So initially we can write here, first tRNA brings methionine on to P site of larger subunit of ribosome. So this is the structural arrangement of mRNA, ribosome, and the tRNA which has already brought the methionine amino acid onto the P site. Now, in the next immediately, this 3'. This tRNA gets separated from the methionine because now methionine has to form a bond, peptide bond, with the next coming subsequent amino acid. Whatever the amino acid may be, it depends upon the codon sequence here. If the codon sequence is cytosine, uracil and uracil on mRNA, then the tRNA which is having anticodon will read that, and if it is guanine, adenine on the tRNA as anticodon, it will read whether there is complementary codon or not. If the codon is okay, then the tRNA will come and fit onto the A site, means amino acylation site. We can also call it as amino acid binding site. So here, according to the codon GAA, that is guanine, adenine, adenine, and the codon cytosine, uracil, uracil, the amino acid that has to be binded on to the 3' end of tRNA has to be leucine. So now leucine and methionine are ready to form a peptide bond now. If you see in the next case, ribosome, both smaller subunit and larger subunit, are moving towards the 3' end from 5'. mRNA will be moving towards 3' to 5' direction. Ribosomal subunits will be moving from 5' to 3' ends of mRNA. So now, this leucine again got separated. Initially, the tRNA of methionine got separated and it was exited from the P site. Now methionine is here. It is forming a peptide bond with that of next leucine amino acid. The tRNA that has bought this leucine also shifted to the P site and it also got separated from the leucine. When any tRNA has come to the P site, then it will get separated from the amino acid. The next one, amino acid codon, if is guanine, 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 then the anticodon on the tRNA will be cytosine, 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 complementary to that of guanine nitrogen bases. When it has come onto the A site, then it will read whether the complementarity is correct or not. Then the amino acid that is coded by this guanine, guanine, guanine codon will be glycine. So the 3' end is occupied by glycine amino acid. So now it is moving from towards the 3' end. One by one amino acid will be separated from the tRNA immediately after shifting onto the P site of the ribosomal subunit. Now, if this keeps on going, then the amino acid sequence will be methionine, as we have written here, which is peptide bond binded with leucine. Next again, amino acid is glycine. Next amino acid codon, if it is uracil, uracil, uracil on mRNA, then anticodon will be adenine, adenine, adenine on anticodon loop of tRNA. Then the amino acid binded on to the 3' end of tRNA will be phenylalanine. So the next amino acid will be phenylalanine. So the amino acids keep on polymerizing to form a polypeptide chain like this. Now we have to discuss one more point. When we have discussed about the complete structure of mRNA, we have discussed that there is 5' methylated guanine cap. This is present on 5' end. So we are calling 5' methylated guanine cap. There are some areas just before the initiating codon AUG. Those are called as UTRs. What are UTRs? UTRs are given full form as untranslated regions. These are present on mRNA specifically just before the place of initiating codon or the translating starting codon area. So this area is called as UTR's place. In the same way, after the terminating sequence, means where the stop codons UAA, UAG, UGA are present after terminating sequence. Here also there is existence of UTRs. Why these UTRs are present before initiating codon, after terminating codon? To give stability to the mRNA molecule. So UTRs are present on the mRNA before the initiating codon, after the terminating sequence. These are useful for giving stability to the mRNA molecule. We have seen in transcription, there is terminating sequence or stop codons to stop the transcription. Then, are there any such types of molecules in the translated unit? It means, yes, when some specific length of the polypeptide chain has reached, then on the 3' end, there are some special structures called as releasing factors. On 3' end, there are releasing factors. These are represented as capital R, capital F, capital 1 because there are 3 types. Capital R 2, capital R, capital F 3. So 3 types of releasing factors are present on the 3' end of mRNA. These two help in recognition of the stop codons, on mRNA end. The third, RF3, it helps in stopping the translation process. It stops translation. In such a way, one complete polypeptide chain or a protein chain is formed by the process of translation.